Cell Structure and Membrane Transport, Part 2. When cells divide, they first need to make all of the components that the new cell will need. In the coming lectures, you will learn how the cell takes in nutrients and breaks them down into simpler building blocks to create those components. This requires transport of nutrients across the cell membrane. Different organisms have different nutritional requirements, and they also grow and divide at different rates. In the coming lectures, you'll also learn how energy and enzymes play a huge role in breaking down nutrients and building cellular components. Organisms take in nutrients from their natural environment. They themselves can be nutrients for other larger microbes, and yet other organisms feed on the metabolic byproducts of organisms they live in close contact with. Eukaryotic cells break down old worn-out organelles and use what they can salvage as building blocks to produce things the cell needs. The cell membrane of all organisms is made up of a bilayer of phospholipids. Recall the properties of the phospholipid molecule that has a hydrophilic head due to the negative charge of the phosphate group, and the fatty acid tails are hydrophobic, or they have no charge. Due to the negatively charged hydrophilic head, the cell membrane is selectively permeable, which means some molecules are allowed entry, but others are not. Charged molecules and ions are repelled by the negatively charged hydrophilic head of the phospholipid when trying to enter or exit the cell, so they must cross through carrier proteins to avoid the negative charge of the membrane. Water molecules, as seen here, enter and exit through protein channels called aquaporins. Most small hydrophobic molecules that have no charge can easily cross the membrane because they are not repelled by the hydrophilic heads of the phospholipids. Very large molecules that are too large to cross the membrane must be transported into the cell by engulfment. Molecules are transported across the membrane either by passive or active transport. Passive transport does not require ATP, which is the form of energy used by cells. Molecules transported into the cell by passive transport are generally small and nonpolar, meaning they don't have a charge. In passive transport, molecules are moving from an area of higher concentration to an area of lower concentration, also known as moving down a concentration gradient. Simple diffusion, facilitated diffusion, and osmosis are all types of passive transport. Active transport requires ATP. Molecules transported into the cell by active transport are generally polar or charged. Their size can vary from small to large. In active transport, molecules are moving from an area of low concentration to high concentration, also known as moving against a concentration gradient. Carrier-mediated transport, group translocation, and bulk transport are all types of active transport mechanisms. Before we talk about transport, let's review solutions. Solutions are made of a solute and a solvent. The solute is the substance being dissolved in the solvent. For example, if you are adding salt to water, the salt is the solute and the water is the solvent. Simple diffusion is the movement of solute molecules from an area of high concentration to low concentration. We refer to this movement as down a concentration gradient, as in the previous slide, movement downhill. This figure shows a solvent in blue we see here the solvent in blue that has a membrane separating the container into right and left sides. Molecules of red dye have been added to the left side of container, and the molecules start to move through the holes in the membrane from an area of high concentration to lower concentration. There is a net movement of molecules until the concentration of molecules is equal on both sides of the membrane, as we see here in this third figure. Keep in mind that when the molecules reach equilibrium, the molecules can now move back and forth across the membrane, but there is no net movement in either direction. If more than one type of molecule is added to a solution, they will both diffuse across the membrane until they are at equilibrium. This figure shows blue molecules were added to the left side of the container, and red molecules were added to the right side of the container. Both types of molecules move down their respective concentration gradients and diffuse from high concentration to low concentration until they reach equilibrium. We know in passive diffusion, molecules move from high concentration of solutes to lower concentration. But some molecules can't easily pass through the selective cell membrane because they are polar or charged. In this case, certain molecules cross the membrane through carrier proteins that span the membrane, so they are not repelled by the hydrophilic heads of the phospholipids. In this example, there is a higher concentration of glucose molecules outside of the cell versus inside of the cell. Most carrier proteins are specific to certain molecules and only transport those specific molecules. For example, this carrier protein is very specific for glucose and will only transport glucose molecules across the membrane. Osmosis is also diffusion, but it's the diffusion of water molecules instead of the solute molecules. Osmosis occurs in cells because of the semipermeable membrane. The membrane stops a lot of molecules from passing through, but water enters and exits to balance out the concentrations inside and outside the cell. A good rule of thumb to remember is that water moves in the direction where there are the most solutes. In this example, the figure on the left shows there are more molecules of sugar on the right side of the U-shaped tube. Over time, water moves towards the side where there are more solutes in order to balance the concentration. Even though the concentration of the solute is equal in the end, as seen here on the right, this changes the volume due to the movement of water. The same thing happens with cells. Water moves by osmosis either into or out of the cell to even out the concentration of molecules between the internal and external cellular environments. The direction of water movement always depends on the concentration of the extracellular fluid outside of the cell, and we always refer to the external concentration when determining the direction of water movement during osmosis. Normally, extracellular body fluids are isotonic to cellular fluid, which means they are the same concentration, so the cells remain the same size. Hypotonic solutions have fewer solutes than those inside the cell, so water moves into the cell where there are more solutes. Hypertonic solutions have more solutes outside than those inside the cell. So water will move out of the cell in the direction where there are more solutes outside of the cell. So you should be able to figure out which direction water will move if given a question with external and internal cellular concentrations of solutes. Osmosis affects cells differently based on their structure. We'll look at red blood cells and compare them to plant cells. In isotonic solutions, as shown here on the left side of this figure, the concentration of solutes inside and outside of cells are the same. So water molecules enter and exit the cell at the same rate. The cell volume and size stays the same in both red blood cells and plant cells. This is great for red blood cells, but plants will appear to look limp and a little dehydrated in isotonic solutions. In hypotonic solutions, as pictured here in the center column, there are more solutes inside of the cell, so water will move into the cell. This is a problem for red blood cells because the cell membrane is not reinforced by a cell wall like in plant cells. Water will enter the cell until it lyses or bursts, but because plant cells have a thick cell wall made of cellulose, the large central vacuole of plant cells fill with water, and plants appear very healthy and firm. In hypertonic solutions, as pictured here in the right-hand column, there are more solutes outside in the extracellular environment than there are inside the cell. In this case, water will leave both types of cells. The red blood cell will become shriveled. The plant cell will also lose water, and the cell membrane may pull away from the cell wall. The red blood cell may not recover if too much damage is done, but the plant cell is more likely to become rehydrated if returned to hypotonic conditions. This figure shows a slide of red blood cells in a hypertonic solution. In this slide, you can see many shriveled cells at the ends of the arrows that have lost significant amounts of water. Here you can see the difference between plant cells in hypertonic conditions on the left and hypotonic conditions on the right. Now we'll talk about active transport mechanisms. In the figure on the left, you'll see the blue transporter proteins that span the entire cell membrane. The molecules that are being transported across the membranes are in green, and the solute binding protein is in tan. In the cytoplasm are sites on the protein where ATP molecules bind to supply the protein with the energy it needs to transport the solutes across the membrane. Because they are moving from an area of low solute concentration to an area of higher concentration against the concentration gradient. The solute binding proteins bind the molecules and carry them to the membrane. Then ATP binds the ATP binding site on the cytoplasmic side of that protein, and then the protein changes shape temporarily to allow the solute molecule to enter the cell. After the molecule moves through the carrier protein, it returns to its original shape, ready to transport the next molecule. Group translocation is another form of active transport. In group translocation, molecules are modified as they travel through the transport protein. In this example, the molecule being transported binds to the protein that is going to transport it across the membrane. As that molecule moves through the carrier protein, it is chemically altered, and this alteration of that solute makes it activated. So the molecule binds to the protein, and as it goes through the protein, it's chemically altered so that once it reaches the inside of the cell, it is active and ready for the cell to use. So by coupling transport with modification of the solute, the cell conserves energy. Although not shown here, the transport protein in group translocation also requires ATP. Larger macromolecules that cannot easily cross the membrane must enter by another route. So, they are engulfed by the cell membrane, wrapped in vesicles, and transported into the cell. This is termed endocytosis. When the cell engulfs large insoluble molecules, we term this transport phagocytosis. The cell can also take in soluble particles that are dissolved in liquid solution, and this is called pinocytosis. Exocytosis is the process cells use to expel or get rid of products. Both endocytosis and exocytosis require cellular energy in the form of ATP. Phagocytosis can be witnessed in single-celled organisms and also in human white blood cells. This figure shows large objects being engulfed by the cell membrane, which also becomes a vesicle. The vesicle, then containing the object, enters the cell. If the substance transported into the cell is fluid containing dissolved substances, then pinocytosis is used, as pictured here on the right. You can click on this video to watch how pseudopods of a single-celled organism performs endocytosis. Exocytosis is the opposite action that allows cells to expel waste products. Everything occurs in reverse of endocytosis. The vesicle with the waste molecules moves to the cell membrane, and then the cell membrane fuses to the membrane of the vesicle. The vesicle then opens and dumps its contents to the outside of the cell. You can click on this link to view exocytosis in action. Use this summary slide to guide your studies on the transport processes outlined in Part 2 of this lecture. This is the end of Part 2. Thank you for listening.